So welcome everybody, particularly anybody who hasn't been before, to a Curious Minds talk or to this lecture theatre. So we're very fortunate to have Professor Kate Jeffrey from the University of Glasgow. Kate's actually a New Zealander, but has been here, there and everywhere work-wise. She's now been in Scotland for some time. Prior to that, she was at University College London. And prior to that, she did her PhD in Edinburgh, so she's used to Scotland and the weather. And I think I will stop there and let her tell you more.
Thank you very much. And thank you everyone for coming. It's very humbling to see how many people want to learn about mental maths. I'm really hoping that I can not disappoint. Claire suggested that I weave in a little bit about myself and my career trajectory into sort of all of science, just because just to kind of make the point that career trajectories can vary. So I thought I would start by introducing myself and saying that I never really intended to be a scientist when I started out. I was going to be a doctor. And what the thing on the left shows is a little snippet from our high school yearbook, where we had to sort of say what we were planning to do and what our classmates thought we would do. And what I was planning to do was medicine, and what my classmates thought that I would do is become an anatomy professor. And that is actually exactly what happened. What happened to me really was that as I was doing my medical degree, I discovered psychology and I discovered the brain, and discovered that there were really interesting and unanswered questions about how it is that the brain can make consciousness and thinking and all of this kind of stuff. And one of the things that really affected me intellectually is that in my final year of medical school, I went hitchhiking around the South Island of New Zealand with my flatmate. And to while away the long evenings on the beach, with nothing to do, I brought this big book with me by Douglas Hofstadter called Gödel, Escher, Bach, which won a Pulitzer Prize for being a really, really incredible and inspirational analysis of how you might be able to get thinking from small units that were interacting. So in other words, neurons, or in his case, because he was a computer scientist, it would have been computer bytes and neural elements. And it was just incredibly inspirational. I just thought, this is the coolest thing. This is so cool. I want to study this. I don't really want to cure people. So I took some time out to kind of think about things. And I went to London for a year. So young New Zealanders will typically, in the course of their kind of education, spend a year or a few years overseas, just kind of discovering the world. And so I went and did some locums as a newly qualified doctor in London. And while I was there, I knew I was interested in the brain, and so I made some inquiries about who I might go and talk to, and someone suggested that I go and talk to this person, John O'Keefe, at University College London. So I fronted up to O'Keefe in his lab, and at that time, so this was in the kind of mid-1980s, and I was in this kind of post-punk phase. New Zealand was a bit lagging in the whole kind of youth culture thing, and I had Doc Marten boots and spiky orange hair, and I kind of went into his lab and said, I want to study the brain. And he said, that's great, let me tell you about place cells. And I thought, okay, that sounds interesting. He told me about place cells. I'm going to tell you about them shortly. And then I went back to New Zealand and I did a master's, and I discovered this structure called the hippocampus, which I'm also going to tell you about, and learned that the hippocampus is really interesting because it's got something to do with memory. And also learned that it's got something to do with navigation. And I started to put two and two together and realised that what I was learning about in New Zealand for my master's had some connection with what O'Keefe was doing in London, but it took a while to bring those things together. Then I decided I really like doing research. I want to keep doing this, and I managed to convince Richard Morris in Edinburgh to take me on as a PhD student. So completely sight unseen. I wrote to him, because that's what you did back in the day, typed a letter, and sealed it up in an envelope and sent it off, and then a few weeks later a letter came back, and I had said in my letter, I'm interested in all sorts of things, interested in the brain, interested in your work, And also I like writing, and I want to kind of win the Booker Prize. And that was the thing that sold me on him. And he said, I like the sound of you, come to my lab. So I went to Edinburgh and studied memory, basically, how memories are formed in the hippocampus. And started to learn and appreciate that an important type of memory that's formed in the hippocampus is memory about where you are. And Richard Morris was actually one of the pioneers of this kind of work. And he invented this way of studying spatial memory called the water maze. He was really, really famous for the Morris water maze. And I was doing some experiments trying to sort of understand how rats... So what happens with water maze is you put a rat in a tank of water and it has to find an escape route. There's a hidden platform somewhere and it has to go and find it. And we were trying to understand, how does the rat know where that platform is? So while I was doing that, it turns out that next door was another lab who was run by a psychologist who was studying learning. And he had taken on a PhD student who had a background in electrical or electronic engineering and artificial intelligence. And this guy, Jim Dennett, was trying to build a robot that could navigate. So Jim and I kind of met one night at the pub, and we really hit it off. I said, you should come and see my rats navigating. And he said, you should help me understand how to get my robots navigating. And so we started talking about that, and we ended up going out. And then we moved to London together. We were seriously going out by that stage. Got married, had a baby, and then it all kind of took off from there. So what happened really is that, so we were both interested in navigation. John O'Keefe, who was a very good friend of Richard Morris, was looking for a postdoc, and he said, do you know of anybody? And Richard said, I've got this person, Kate. And John remembered me. And meanwhile, John's collaborator, Mike Recce, was developing computer simulations of navigation, and he needed somebody with some computational and electronic experience, and so he hired Jim, and so we both moved to London together. And that was just really completely fortuitous, but we just found ourselves actually at the beginning of what was really a revolution in psychology and neuroscience, because O'Keefe had made this incredible discovery, which he eventually won the Nobel Prize for, about place self. So it was a very, very intellectually fertile time for us. We also had a baby, so it was more than just intellectually fertile. This one's now a patent attorney. They're all quite grown up now, and they're all doing their own thing. So what I want to do is to talk a bit about those discoveries. So Jim and I contributed a little bit to those discoveries. We didn't quite get the Nobel Prize, but we certainly had a front row seat onto these discoveries. And what O'Keefe really did was unlock the map and the brain, essentially. So let's just talk a bit about maps. So we use maps to find our way around. So if you need to go somewhere, you get your phone or something, and you call up Google Maps, and you have a kind of a plan view of where things are. And that's a technological advance. But we've been navigating for a lot longer than that. And traditionally, we have navigated the way that humans have navigated for millions of years, and animals navigate too, which is we don't have maps. We move through the environment and we see what's around us, and somehow our brains are tracking what we're doing, and they're storing that information as memories, so that when you're back in that place, you know where things are and you can compute your way back to it. It's really a remarkable process when you think about it. And the question is, how do we do that? And it's not just navigating across the desert or large spaces. We also navigate around our own homes. Like when you're in your home, this isn't my home, that's just a random home from the internet, but it's quite a large home. My home's not this big, but as you're navigating around, you know where things are, right? You kind of know when you're in the kitchen, you could point to where the hall is or where the bedrooms are. So you have a kind of map of the space around. And the question is, how is that map formed? So much of our understanding of how we work and our own brains work comes from studying animals, and particularly rodents. Rodents have been a really important kind of substrate for understanding the brain, because they're mammals, they're vertebrates, they're mammals, they're quite similar to us in many ways. They're social. Rats are quite smart. Mice are really dumb, but they're kind of, they're useful in other ways. So mice are not actually small rats. They're really very different, but they're very good for studying. They've been bred for many generations to be. We've kind of domesticated them, so we can work with them in the lab, and they're fairly docile. They're unnaturally docile for wild animals. So a lot of work has been done with rats, and psychologists get a lot of stick for studying rats in mazes, but actually that's told us an incredible amount about how things work. And one of the big advances in the last century was that psychologists were trying to understand how rats find their way around mazes. So they would put a rat in a maze, and they would note that the rat would explore, and then it would discover food. And then if you did that a few more times, you put the rat in, and it would find its way straight to the food. And there was this very elaborate theory of how this worked, which we call associative learning, which is that the rats were learning, okay, if I run straight and then turn right and then left and right again, I'll get a reward. And so the idea is basically that the animals are just forming associations between an action and a reward, and that's what they're learning. And then this guy, Edward Tolman, said, I'm not sure that that's the whole story, because I can see rats doing something that's not explainable that way. And his most famous experiment is this sunburst maze experiment. So he would train rats to come from here, run down this corridor that's got a bit of a bend in it, and find food. And they would do that over and over until they were really good at it. And then he would do what we call a probe trial, which is to try and find out what the rat knows. And he blocked off the arm, this one, that the rat normally would run down, and gave it a whole lot of other choices and said what the rat's going to do. And he found that what they tended to do was to run down an arm that was in the direction of the food, even though they'd never done that before. So that's quite hard to explain on the basis of the rat had learned an association between an action and a reward, because they'd never done that action. And he said that he thought there was a map in the brain of the rat. He called it a cognitive map. And he said, we believe, this is the quote down here, we believe that in the course of learning, something like a field map of the environment gets established in the rat's brain. So this was a tremendously unpopular idea, because people said that the idea that there's a map in the brain is just ridiculous. How would you do that? And there's no evidence for it. We can explain pretty much everything with this stimulus kind of response learning. But the idea was there in the literature, and then it got resurrected in the 1970s. And the reason it got resurrected is because of my friend John O'Keefe, who's a lot older in this photo than the one I just showed you. This is much more recent. But O'Keefe had moved to London in the late 60s, early 70s, and he brought with him from the States a way of recording brain signals from rats that were moving around. So nobody had really done that very much before. When neuroscientists had started to record from living brains, they were from anaesthetized animals. So you would anesthetize the animal, make a little hole in the skull with a scalpel and so on, and put an electrode in and record the neurons. But that didn't work in animals that were moving around. So scientists started to develop that technique so that you could record from animals that were moving around. So they used wire electrodes instead of glass electrodes. And so what you could do is implant a little wire into the rat's brain under anesthesia. It would wake up. It had no idea that this had happened because there are no nerve endings in the brain. But you could connect that wire with a little connector to a recording system as the animal was kind of doing whatever animals do, in this case wandering around looking for food. And you could detect the activity of the single neurons while the animal was doing stuff. It's a really, really incredible technique. It sounds a bit gruesome, but from the point of view of the rat, it doesn't really know what's happening, it's just getting food. From the point of view of the scientist, it's reading the rat's mind, essentially. It's the most incredible experience to be able to listen to the language of the neurons. And he was recording from the hippocampus, and I'll tell you why in a second. And he would just do something really simple, which is to get the animals to walk around on this sort of platform. He would take the signals from the electrodes, feed them through an oscilloscope and a computer and so on. And you'd see a signal like this, which is just a typical oscilloscope trace. These little jagged things, these are spikes, these are nerve impulses, action potentials from a single neuron. So as the rat's walking around, the neuron that's right next to the electrode gets excited and starts firing nerve impulses, and those are detected, and you can feed them through a loudspeaker and listen to them, or you could feed them into your computer and then plot them spatially to see where they were and so on. And he was listening to these things, and he noticed that when he was listening to one of these neurons, it would become really, really excited every time the rat went into one particular place in the environment. So the rat's walking around all over the place. You can see from the schematic, it's kind of wiggly path. But every time it goes into this corner over here, this neuron, whose actual potentials are shown, would become really active over here. And he recorded a few of these neurons, only about 17 or so, a really small number. But it was the same thing everywhere. Like, each neuron had a place in the environment that it liked to be active. And it didn't matter whether the rat was walking, this way or that way. So it didn't matter what it was looking at. It was when the rat was in the place that it would become active. And O'Keefe named these cells place cells. And his argument was they are responding to the place. They're not just sensory cells. They are doing something more abstract. So this is the schematic that I just showed you. This is an actual place cell that was recorded with a camera that was looking down, and the rat was walking around all over this square box, and this is the actual potentials from that one neuron. So that's the activity of one single neuron. I actually have a recording of a place cell, and I'm not sure if it's going to work because I'm not sure how our sound is, but let's try and see. Okay, I don't seem to. Okay, it's not going to work. Sorry about that. It's not actually activating the video for some reason. Anyway, if you heard it, you would be amazed. Trust me, it's amazing. You can actually see these things online if you do a search, and it's really worth listening to, because when while I was in Richard's lab, I went down to John O'Keefe's lab to visit, and I saw my first place cells, and then I just fell in love with them and thought, I want to study these things. So after my PhD, I went, as I said, and got a postdoc with O'Keefe. So I said he was recording from the hippocampus. So this is the hippocampus here. This is a rat brain. These are not to scale, obviously. This is a rat brain, this is a monkey brain, and this is a human brain. And these coloured areas are the homologous areas in those different brains. So the yellow is the same structure in each of those three species. And you can see that the shapes and sizes are a bit different, but the arrangement is more or less the same. And actually the interconnectedness between them is all the same. So the last common ancestor of rats and monkeys and humans was like 90-something million years ago. So in that 90 million years since, we've all held onto our hippocampuses, all these species. So that's telling us that it's quite important. In the rat, it's a really large structure because rats don't have very much of the cortex, the outer wrinkled part of the brain that humans have a lot of. Monkeys are kind of in between. And in monkeys and humans, the hippocampus, it kind of lies down, flatter, more horizontal. In rats, it's a little bit more vertical. And I'm showing you that because we're going to look a little bit more at rat brains and human brains, so just so that you can see what these are. So O'Keefe was recording from a rat, so he was recording from here. And if you take a cross-section and look, so here's the rat brain, here is the hippocampus. If you take a cross-section through the brain, this is what it looks like. And this area here, it kind of looks like Swiss roll. It's quite a very, very striking pattern. Those are rows of neurons all packed together. So the hippocampus has really, really huge, densely packed neurons. It's a very busy structure. If you use modern genetic techniques to make the neurons all different colours, and then have a look at them with a fluorescent microscope, you can see that it's a really intricate and beautiful structure. So there's a row of cells like this, which we call the cornu aminis, CA, and there's another row of cells here called the dentate gyrus. And these two interlocking things are, although it's quite small, incredibly important for our sense of not just where we are, but as we'll see, also for remembering the things that have happened to us. So O'Keefe was recording from the layer up here. So the electrodes had gone down through the cortex and they were sitting in that cell layer. So that's what he was recording from. And that's mainly where we see these place cells. So in the dorsal, the top end of the hippocampus. So humans have them too, and it's a little bit harder to to find them in humans, because we're unfortunately not allowed to just kind of drill holes in people's heads and put electrodes in. But actually, you are allowed to do that under one condition, and that's if the person has epilepsy and it's not responding to medication, and they're going to need surgery to destroy the damaged bit of brain that's causing the problems. So temporal lobe epilepsy is a problem in the hippocampus, and neurosurgeons these days, in the old days, and I'll come back to this, but in the old days they just used to carve out the hippocampus with, just excise everything. That turned out not to be a really great idea, so what they want to do is only carve out the important bit. And to find out the important bit, they put electrodes in to find out which is the hyperactive part of the hippocampus that's firing off too many action potentials and generating seizures. So while these patients are in hospital with their implanted electrodes, neuroscientists can kind of come along and say, do you mind if we just have a little peek at what your neurons are doing? So they will get, obviously this is carefully planned and there's a lot of ethics and all this, but they will get the patients to explore, and they can't explore physically because they're in bed, but what they can do is explore virtual reality. So here's a kind of a virtual reality environment. So this person was sitting in bed with a computer and a mouse, and they were exploring this town, and it's a little bit hard to see because it's quite small, but from what you call the first person perspective, this is what they can see, buildings and streets and so on. If you look down on the town from above, it's actually a kind of a pretty simple town with a grid of streets, and there are different scenes on each of the streets. And when you get people to navigate around this very, very simple town, as if they're having to find their way around, so they're told, can you drive to the bakery? So they're driving an imaginary taxi. Can you take us to the bakery or whatever? If you record neurons from these electrodes that are implanted, you find that there are places in that virtual environment where a given neuron will become really active, so there's a hotspot of activity. So these are place cells. We found quite a few of them now in humans, so we're pretty convinced that we are pretty much like rats in the sense that we have hippocampus and we have these place cells. So why was O'Keeffe interested in the hippocampus? Well, the reason is because of this very famous epileptic patient. This is a young guy at the time called Henry Mollison, who developed really, really, really bad temporal lobe epilepsy in the 1950s. And back then we didn't have all of these sophisticated techniques. We just had surgical excision. So what surgeons would do is to excise that whole temporal lobe where the hippocampus is, and that would usually cure the seizures. And it didn't seem to have very much effect on people afterwards. They seemed to be kind of normal. This guy was slightly unusual because he had a focus on both sides of the brain, and the neurosurgeon, William Scoville, decided to excise both sides. He said, we think the temporal lobe is a silent area of the brain, not sure what it's for, we don't use all of our brains or whatever. So he excised both temporal lobes, so both hippocampi and some other things as well, got taken out. So you can see here, this might actually be a slice of HM's brain. So he used to be called HM for many years until he died and they could release his name. So the older literature you'll see HM. So he had his hippocampus removed on both sides. And when he woke up, he had no more seizures, so he was cured from his epilepsy, but he was completely amnesic from that moment forwards. He was unable to form new memories. And it was a very, very striking and profound amnesia. And he ended up spending the rest of his life in an institution. He was unable to function in the real world. It's an extremely tragic case. But he's been absolutely foundational to our understanding of how the brain makes memories. Because this case, which was immediately written up, so Scoville and Brenda Milner, who's a neuropsychologist working with him, they immediately published this and said, don't do bilateral resection of the hippocampus. This is a memory structure. We didn't know this. And meanwhile, neuroscientists said, this is a memory structure. This is interesting. We didn't know that this was a thing. So they were interested in the hippocampus, and that's why O'Keeffe wanted to record these neurons from the hippocampus. The other notable thing about the hippocampus, which many of you might know, is that it's one of the structures that degenerates particularly prominently in Alzheimer's disease. So Alzheimer's disease eventually affects the whole brain. This is a comparison of a normal brain on the left and the brain of a patient who died of Alzheimer's disease on the right. And you can see that the whole brain is quite atrophied, it's very shriveled. But the hippocampus is particularly badly affected. That's it there. So here's a normal one that's quite looking plump. And here it's kind of more or less almost disappeared. Alzheimer's disease starts in the part of the brain that feeds information into the hippocampus and spreads. So it spreads down into the hippocampus and then spreads from there to the rest of the brain and eventually takes over the whole brain. And we now know, because of the work of O'Keeffe and others, that one of the very first symptoms of Alzheimer's disease is getting lost. It's often the first thing that people complain of. They went round to the shops, they'd lived there all their life, they know where the shops are, but they couldn't find their way back, they got lost. And then eventually it spreads until they become unable to remember the events of daily life. So they initially lost their ability to find their way around, their map of space, but then they lost the ability to remember the things that happened in those places. So that suggests to us that the hippocampus is making a map of space, and that map is being used to organise our memories. It's linking to the memories. So first you lose the map, and then you lose the memories, basically. You don't just lose memories, but you also lose the ability to imagine new experiences that you've not had that are spatial. So some work that was done by Eleanor Maguire at UCL with patients with hippocampal damage. So they had hippocampal removal or hippocampal damage or maybe early Alzheimer's, so various types of damage. She got them to kind of imagine scenes or to imagine other things and then describe what they could see. And she found that the people with hippocampal damage, if you said, imagine you're on a beach, standing on a beach, what can you see? And a normal person would say, okay, well, I'm standing here, I'm on the sand, I've got the sea to one side, there's palm trees, off in the distance I can see an outcrop of rocks and so on. They couldn't produce that integrated, kind of spatially organised scenario. They knew the elements, but they couldn't put them together. It's like they just had nowhere to put these things from their imagination. So this was much more recent, but going back to O'Keeffe's discovery of place cells, so this was the early 1970s. So he and Lynn Nadell, who's a neuropsychologist that he was working with, proposed that what the place cells are doing is implementing this cognitive map that Tolman had suggested that rats have. So Tolman's work was psychological. He showed that rats could find their way around. O'Keeffe and Nadell said, we think that the hippocampus is the site of this map. And they wrote this book, which was about as controversial as Tolman's proposal. It didn't really land all that well in many quarters because it, again, violated this doctrine that the brain doesn't make internal maps. It just didn't, people just couldn't really relate to the idea. They couldn't imagine how this could work. So the book wasn't all that well received at the beginning, but then it became more and more popular. And now I think it's a kind of collector's item. This photograph is my copy of it. And when O'Keeffe won the Nobel Prize, I got him to sign the copy. So that's me at the Nobel champagne reception on the left, getting him to sign the book. And then a few years later, when Richard Morris retired, he invited Lynn Nadell to his Festschrift, his kind of retirement symposium. And I knew that Nadell was going to be there, so I brought my book along and got him to sign it as well. So this is my most treasured possession, the double-signed copy of the hippocampus as a cognitive map. So how can the brain make a map? Like, how could this work? So we need to kind of think about how does a place cell know where it is? This rat's kind of walking around this platform, and every time it goes to a particular place, the cell becomes active. How does that cell know where the rat is? So there's two ways of thinking about the answer. One is, what is the information? Like, what can rats see and hear and smell and so on? What's the information coming in through the senses that's finding its way to the place cells? And then the second thing is, what path does the information take to get there? So it's kind of the same question, but one is a kind of an anatomical question, one is a kind of a psychological question, but it's coming to the same thing. These cells are deep in the brain. There's 2 or 3 neurons between the senses and the place cells. So what is that information that's coming in? So when I went down to O'Keeffe's lab as a postdoc, I started recording place cells, and I started playing around with the sensory environment to try and get a handle on this. So this is one of the experiments that I did, and I'm presenting my experiment because I can. But actually, there's there's a huge amount of work that was starting to be done. So what I'm presenting is an example of the type of finding that was emerging from people's labs at the time. And I'm showing the place cell not as a bunch of little red dots, but now as a heat map. So this is the more modern way of showing the activity. You kind of average the activity of the cell and say, where is it most active? And you get these kind of blobs. It's just a little bit easier to kind of work with when you can see it like this. So what I did was I recorded place cells in a square box. So, about a metre kind of square with walls about so high and curtains around the outside so that the rat couldn't really see which direction was which, other than that there were 4 walls around it. So from the point of view of the rat, it was in a box, and the box kind of looked the same in 4 different directions. So to help the rat and the place cells be oriented, I would put a cue card, what we call a cue card, which is basically a big piece of white cardboard with a spotlight on it. I would hang that from the curtains so that the rat could go, okay, I know where I am because that's the big card over there. And you can, if you do this carefully, you can record the rat and the place cell. You can take the rat out. You can move the cue card around, move nothing else, just the cue card, and you put the rat back in again. And because the box is square, the rat doesn't, and because the rat was a little bit disoriented when you took it out and put it in a box or did something with it, maybe wiggled the box around a bit. When you put it back in the environment, it goes, oh, there's the cue card. I know which way around I am. So if you record a place cell while you're doing that, you can see that the place cell has rotated by the same amount that the cue card rotated. So the cue card went from the north wall, let's call it the north wall, to the west wall, and the place cell has gone from the southeast corner to the northeast corner. I always have to stop and think about that. Northeast corner. So if you imagine the rat thinking the east is over here, it's as if it's thinking, oh, I got that wrong. No, that's something else. Forget the arrow. The one other thing to note, though, and I don't know if you can see it because it's not really good contrast, but there's a little X in the corner here, and this is because the rat was quite used to the environment and it would get a bit bored. And when it was bored, it would curl up in a corner. And what I found was that the corner that it curled up in rotated along with the place cell. So we're seeing not just what does the place cell think is the right corner for it to be firing in, but also what does the rat think is the right corner for it to be sleeping in. And you can control it with that cue card. So wherever the cue card is, the place cell is firing on the opposite wall and the rat is in the nearest right-hand corner. So we seem to be controlling the rat's internal sense of direction, if you like, with this cue card. So that's telling us that vision is one of the things that's coming into the place cells. They're somehow knowing what the rat can see. But what you can then do is take the cue card away. You can record the place cell, and there'll still be a nice, we call them place fields, these blobs where the cell is active. So even though there's no cue card anymore, the cell will still fire in this nice kind of condensed area. What I then did is I took the rat out, and instead of disorienting it, I put it in a box, and I very slowly rotated the box so that the rat didn't know that it was rotated, and then I put it back. And I found that the place cell rotated by the same amount that I had rotated the rat. So the internal sense of direction of the rat was enough to tell that place cell where it should fire. So that's a different type of information. If you've got a cue card, that's vision. If you are relying on your internal sense of direction, that's what we call self-motion tracking sensors. Sometimes it's called idiothetic. It's kind of the sensors that your brain uses to know how much you've moved, even though you can't see. So if you close your eyes and move around, you kind of know which way you're facing because your vestibular system, your inner ear accelerometers, tell you how far you moved. Your motion command center said, I've told my body to move 90 degrees and so on. So there's all this other information. So we know that the place cells are getting that as well. So they're putting all of this together. They have multiple sources of sensory information. The other way of looking at this is what is the path of information coming in. So that this is the hippocampus here. So here's a very, very simple circuit diagram. So here's the hippocampus. One of the most important inputs to the hippocampus is this area here called the entorhinal cortex. So here it is in green. this is an area that's collecting all sorts of information from other parts of cortex. So, for example, the visual areas of the brain, the auditory areas of the brain, the somatosensory, touch and so on, those areas of the brain. All of that's going into the entorhinal cortex and then into the hippocampus. So it's getting huge amounts of information. It's also getting a lot of stuff coming up from the deeper parts of the brain. And so all of that self-motion information that I was telling you about, that's all coming up through the brain stem, through the vestibular system and so on. So it's all of this sort of stuff happening. And it's not just what can you see and where do you think you are, but it's also things like how do I feel. So there's a lot of emotional inputs and so on. So the hippocampus is just getting area. It's just lots and lots of stuff. So that makes it quite tricky to study, I have to say. So this sense of direction that I was manipulating with my rotating things comes from these neurons called head direction cells, which are found in these areas outside the hippocampus. A head direction cell will fire when the rat's head points in a particular direction. And you can think of it almost like compass needles. So here is a schematic of a whole bunch of head direction cells. They're color-coded according to the direction that they're facing. And so you've got a different cell for every direction. So as you're kind of moving around, some cells will stop firing and other cells will start and so on. So you have this kind of compass in your head. And what the brain has to do is to learn the association between this internal compass and the external world. So when you go to a new space and you see the external world, you have your internal compass, you have to kind of line it up and learn what the kind of mapping is. And then the next time you go into that space and see those now familiar scene, that can tell your head direction cells, okay, you should be facing this way. You want to fire this way. So the brain is trying to link the internal map and compass with the external world and remember those links. So memory is very, very important. So here's a little schematic of a visual scene which you might recognise. So there's a kind of a compass, but the other thing about the place cells is that they fire. They don't just fire in the right corner, but they fire at the right distance from the walls of that box. And so there's something that's telling them how far the rat is away from the walls. And it wasn't for quite some time that we started to get a handle on how that information comes in. And this experiment was done by this couple here, the Mosers, Edvard and Margaret Moser. They're a married couple who came to visit the O'Keeffe lab when Jim and I were there, and we taught them how to record these single neurons, these place cells and so on. Biggest mistake of my life, because they went back to Norway. They set up their new lab. They were quite young scientists like we were. They set up their new lab. We'd had some sort of conversations, and we all agreed that what we needed to do to understand the place cells was to record from this entorhinal cortex area. So they recruited a couple of very talented PhD students, Torkel Hafton and Marianne Fern, and they figured out how to record from the entorhinal cortex. And it's not easy, I can tell you, because I was trying to do this too. And I was getting neurons that I was struggling to make sense of. They persevered and persevered, and following a tip-off, and I won't go into the details for time reasons, but they kind of had a tip-off from the scientists at a meeting that maybe they should record not in a normal-sized box like we normally would use, but in a much bigger one. And so they recorded in a much bigger box, and they recorded these neurons in entorhinal cortex, and they found something really, really extraordinary, which is that these neurons also fire in restricted regions of the environment, so kind of like place cells. But whereas a place cell usually has kind of one or maybe two or not very many places where it would fire, these cells would have multiple locations. So they'd fire all over the place. And not only that, but whereas the region where a place cell would fire might be quite sort of elongated or sort of strangely shaped, these cells would produce these very nice circular firing fields. They really produce these compact firing fields. And the most extraordinary thing is that the spacing between these patches was very, very regular. So this, what you're seeing here, is one cell. It's not lots of cells, it's one cell. And it's firing here. It's firing when the rat goes over here. It's firing when the rat goes over here. And meanwhile, you can see the rats walking all over the place. So it's not a simple job for the cell to figure out that the rat is here, or it's here, or it's here. So it's just producing this really, really incredibly regular array. And if you look at these firing fields, they kind of line up in these rows, which have a particular orientation which we know is controlled by the head direction system now. And if you look at each one of these fields, it has 6 surrounding it. So there's a kind of a hexagonal pattern. It's actually a pattern that physicists and mathematicians call hexagonal close-packed array. So I remember the moment I saw the paper with this, and I remember my jaw dropped. I think I let out an expletive of some sort. It was like, this, I cannot believe what my eyes are seeing. This is extraordinary. And we all rushed off to the pub to pore over the paper and try and figure out what on earth was going on. And so this discovery was the other half of the Nobel Prize. So O'Keefe won half the Nobel Prize for place cells, and the Moses won the other half of that Nobel Prize in 2014 for these grid cells. The really annoying thing for me is that when I go back and I look at the recordings from entorhinal cortex that I'd been trying to do, I had seen a grid cell, but because I was recording in a little box, I hadn't recognised this big pattern. It's just a kind of a small pattern. But anyway, it's a very exciting finding, and the reason it's exciting is that the even spacing tells us that something is tracking the distance that the rat has walked. So you've got a kind of an odometer in your entorhinal cortex, and it's producing this thing that kind of looks like a grid, so that's why they're called grid cells. And our immediate assumption is that it's kind of like the grid reference on a map. It's marking out distances so that the place cells can use them to figure out how far the rats walked and therefore where they should become active. So we had a window seat into these discoveries. It was a very, very exciting time. So we have these 3 types of cells that have been really important for understanding how this map in the head is kind of put together. I should say that it's really only the tip of an iceberg. So I got my postdoc to do a trawl through the literature to find out actually how many different kinds of cells there are. And there's many, many brain areas that have cells that are doing something a bit mappy. So we've got the place cells, the grid direction cells, the grid cells, the head direction cells. I didn't talk about these ones. These are border cells. These fire when the rat is up against a wall. There's just all sorts of stuff all over the place. There are some cells now that fire when the rat is at a particular distance and direction from a wall. So it doesn't matter where it is, but if there's a wall at a particular distance and direction, that cell will fire. It's just amazing, all of the different signals that are out in the brain. So it's all being pieced together so that you can figure out where you are. So the map, we're still trying to understand like what reads the map. How is this information used to give you a sense of space? But we're slowly getting there, and we've learned a few things from these neurons. One thing that we've learned by studying the place cells is that place cells get quite confused by repeating spaces. So, this is quite common in our built environments, is that we have identical rooms, identical offices, corridors with lots and lots of doors, and it's very easy to get confused. So we've done an experiment where we record place cells in kind of repeating boxes, so they're corridor with like little rooms. And we find that the place cells tend to fire in each of the rooms, so they don't seem to be able to realise that this is the first room, and this is the second room, and this is the third room, and so on. So it's easy to fool a place cell in a rat. We humans were pretty smart, and so we can use other things to help us, like we know that there's a number on the door that's telling us what room it is. We recognise person A's furniture from person B's furniture and so on. So we don't get completely confused, but it is the case that repeating structures in architecture can be confusing, and I spend a lot of my time talking to architects and trying to convince them to not do this, unless they want people to be confused. The other thing that we've learned is that the head direction cells, so still haven't fully nailed this down, but my kind of hypothesis, belief, is that to really have a good understanding of a space, so thinking of this building, for example, what you want your head direction cells to do is to have the same firing directions in one room as they do in the adjacent room. So if I'm a cell that likes to fire when I'm facing this way in this room, I would like that same cell to also be firing this direction when I'm in the next room, so that there's a coherence between the rooms in the firing directions. So I believe that that is important for making an integrated map of a multi-room space. I'm trying, in the process, trying to prove that, so as some other people. We haven't quite nailed it down yet, but it seems very likely. And one of the reasons that we think that is, in fact, recordings that have been made where you've got head direction cells in one room and then the rat can walk under its own steam into the adjacent room, so like you walk out of your kitchen into the hall or something like that. The head direction cells, provided you don't disorient the rat, the head direction cells will maintain that orientation. And they can do that because of this self-motion tracking that I told you about. So even though they go into a new room and they haven't seen that room before, so they can't use vision, they can use those other things to go, this is, you're facing the same way here as you were there. And then they can learn the visual, the scene that matches that direction. And then subsequently, when they go into that room, they can use that visual scene again. So there's this interplay going on. So we think that there needs to be linking information. And again, we're trying to tell architects, please can you make it easy for that to happen? So it's quite difficult if you go out of a room and round a corner into the next room, or into a stairwell that's got no windows, and you're going round and round and round, you get very disoriented and so on. You really, ideally, you want to help the head direction system be able to make this coherent representation with windows or spaces that you can easily see from one to the other, or some big landmark that you can see from any of the rooms. I've wondered whether we could help in complex buildings by having something like paint the north wall always a darker colour than the other walls, or something like that, so that you could use that. I don't know. So there's a few things to think about there. The other thing that we've learned is that the head direction cells in rats, they set up their firing directions within seconds. So if you put the rat into a new environment, the cells are immediately active. And then if you take the rat out and put it back, then they'll take up that same firing directions. So if the rat had walked into that space, then the direction that they're active will, as I just said, match the directions that it had in the previous room. If you disoriented the rat, and so it didn't know which way was which, then the cells just guess, as far as we can tell. They just go, we don't know where we are, but let's just fire this way. And then the next time the rat goes into that space, they'll fire that way again. So if you put the rat into adjacent rooms, but they've got different firing directions in those rooms, it's going to be hard for the rat or the person or whatever to make an integrated map, because it's got conflicting directional associations with those spaces. And this is a really common experience for us in some of our spaces. So, for example, you probably, have all had the experience of travelling on the underground, and you come out into a space and you don't know which way you're facing, but you kind of form a kind of directional impression, and then you kind of walk around and you realise actually this part of London or whatever is adjacent to a bit I do know, and in this space I feel when I'm facing this way, this is a particular direction, maybe north. Over here I feel like that direction's that way because I set myself up wrong the first time I emerged into that space out of the underground. That happened to me actually when I got to Edinburgh. When I arrived in the city, I somehow got south and north mixed up. And in the 3 years that I was there, I never quite managed to untangle them. I always felt that the river was to the south, whereas I think it's the north, isn't it? See, I can't remember. I was always confused. And so I have this belief that to help people navigate around a space, we need to help them make a mental map, and to do that we need to give the head direction system information that they can use. Another thing that's really complicated for head direction cells is if the space has a rotational symmetry, it looks the same in different directions. That can be really confusing. It's not confusing though if there's a mirror symmetry, because although it has a nice pleasing symmetry, which architects seem to like, it doesn't confuse your head direction system. There are kind of places where the visual scene looks exactly the same, even though the facing directions are different. So some types of symmetry are okay, some types of symmetry are not okay. And then finally, for head direction cells, the cells prefer distant landmarks. So to the extent that they're using the visual scene, they work better if the landmarks are far away, because they will maintain a constant directional relationship to you, whereas if you got something that's close up. So for example, if I was trying to use that screen as a directional landmark, the direction that it is relative to me varies depending on where I am in the room. So I couldn't use that to tell me which direction is which in the room. All the directions in the room kind of point towards the screen in different angles, if you see what I mean. Whereas if you've got something that's really far away, it's a constant direction. It's kind of parallel directions. So some experiments with rats have suggested that head direction cells, that system prefers to use the more distant landmarks. So I'm going to wrap up because time's running on, but I just wanted to finish by answering the question about why we vary in our spatial abilities and to say we don't yet know, but we do vary quite a lot. So I've been doing some experiments with humans using virtual reality, so walking around buildings, and what I'm getting them to do is to walk around these buildings with different structures and then draw maps. And some people are quite good at drawing maps, so here's quite a good representation of this space here that they explored. This person's trying to draw this map of this space, and you can see that it's a much less good map. In fact, this person has imposed a mirror symmetry on a space that actually had a kind of a rotational symmetry. So they've used some type of inference to kind of help inform their mapping. So they kind of lost track of what they were doing somewhere along the line. We also know that there's different ways of solving spatial tasks. So I've been talking about the hippocampus and its map-based space. And we use the hippocampus in situations where we need to think about our navigation. So particularly when you're in a new space and you don't know where things are. But once you're really familiar with the space, it becomes automatic to navigate. So this kind of illustration is meant to show your commuting route. And let's say you're commuting to work and you always go down this road and you know which way it is and you don't really think about it. And you're not using your hippocampus anymore. You're using a structure called the striatum, which is involved in habit-based actions. So when you're on autopilot, you're using your striatum. When you're thinking about it, you're using your hippocampus. So you may be on autopilot commuting to work, but then you come across roadworks one day and you go, I can't take my normal route, I have to replan my route. What would be the closest, the shortest route? I could go down that street and then I'd have to go down blah, 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 and so on. So you're calling to mind a mental map, and that's when you're using your hippocampus again. And the extent to which people naturally use their hippocampus or naturally use their striatum and base their navigation on cues in the environment varies between individuals. So some people are more likely to anchor their actions to the environment, other people are more likely to use a kind of a global sense of direction and so on. And the other thing is just that the brain is very complicated and different people have different skills, if you like. Some people maybe have a really, really good somatosensory association cortex, or maybe really good entorhinal cortex, the grid cells work really well, or whatever. What's not mentioned here is a kind of cortex called retrosplenial cortex, which my lab is really interested in, and we think is the part of the brain that's involved in looking at landmarks and figuring out what direction they are indicating. And there's some evidence that people vary quite a lot in how much their retrosplenial cortex does that. And then finally, there's a question that I get asked a lot, is it a case of use it or lose it? Like, are we losing the use of our hippocampus with our smartphones and all of our navigational aids, and we no longer have to think as much about navigation anymore? And we don't really know. I think it's fair to say that the data are not in yet because we haven't had smartphones for long enough. If there is an effect, it's probably one that takes many, many years to develop. There is some evidence that the hippocampus is plastic, so some really, really interesting work by Eleanor Maguire, who's been really a pioneer of studying the human hippocampus in navigation. So she did some very famous work studying London cabbies who spend all day long navigating, so they are extremely good navigators. And she did a study to look at the size of the hippocampus in these people, and what she found was that the part of the hippocampus which is the equivalent in humans to the part where O'Keeffe found the place cells, so that's the kind of the top part of the hippocampus, and the rat is the back part in humans. She found that was enlarged in the taxi drivers relative to non-taxi drivers, and it was more enlarged the longer they had been driving. So there's some suggestion that the hippocampus was kind of enlarging with use. So we don't know for sure, it's been hard to replicate and so on, but we do know that the hippocampus has a lot of plasticity, a lot of new connections. So it's a really busy structure, as I mentioned. It's busy forming memories and so on. So it's quite possible that as we change the extent to which we use our hippocampus, we might be having effects on our brains we don't know. And then this is almost my last slide. Recently, people have started to speculate that in humans, we don't just use our hippocampus for navigation and for episodic memory, like life event memory. We might use it for reasoning in a domain that's not actually spatial, but you can think of it spatially. So, for example, thinking of a social network, thinking of all your relatives. You've got parents, you've got cousins, you've got aunts and uncles, you've got grandparents, you've got and maybe you could widen that and go there, all their friends, and so you know a huge amount about social relationships. And there's some evidence that hippocampus might be used to represent those things. And so that's one of the really exciting future things that we're starting to look at is the hippocampus for more than just space. So this is just the take-home message: look after your neurons, because they're really important, and we can't do without them. So thank you very much for your attention, and really happy to take questions.
Questions, please. Lady in blue.
When you were talking about navigating our environment, it came to my mind it's similar to feng shui, which is the Eastern process of making your environment, moving it. And I know that architects use concrete in buildings. Is there any connection?
So the architectural practice of, I don't know if I'm pronouncing it right, feng shui, is that right? And I don't know that much about it, but it involves an importance of the positioning of the objects in a room for its effects on your psyche, basically. Is that a fair summary? And would that use the hippocampus? So, yeah, quite probably, because there are neurons, I showed you that picture of all the many different kinds of neurons, and there are neurons that are sensitive to the location of objects in a space. And interestingly, there are also some neurons that are sensitive to when you remove that object. They're still active to where that object was, so they remember where it was. And like if you've got a cat and you move something in the room, you move a box or something, the next time the cat comes into the room, it goes and it checks out the box because it knows that it's moved. So the system is very sensitive to the location of things in it. So yeah, for sure. Do you want me to just? Yeah. Okay.
Thank you very much. I mean, very interesting talk, fascinating. What I was wondering was if you spoke quite a lot about visual perceptions. If the visual perception for some restriction of the person isn't available, then what skills are they using for the mental mapping? And taking that further, are there other perceptions that people, such as magnetic or gravity or some other, we know that birds and certain animals have those perceptions to use their mental mapping. Have you looked into that, and does that feed into the hippocampus?
So the question is, are there other senses other than vision that feed in, like magnetic sense that some animals have, and so on, and you could, and from that you could say, if somebody has no vision, if they're blind or if they've got their eyes closed or something, can they make mental maps? And the answer is yes, they can. And the reason is that the hippocampus is getting lots and lots of different sources of sensory information. So one of the important ones is this sense of self-motion, and that'll tell you how far you've moved and in what direction. But you need to anchor that to the world. And when you've got vision, you can use vision to anchor it, where you are. If you haven't got vision, you can use touch. So an experiment with rats that were deprived of vision found that the place cells were perfectly normal, but they wouldn't start to be active until the rat had bumped into an object and it knew where it was. So then, having kind of set its starting point, if you like, it could then keep track. And every time they bump into the walls, that kind of resets the system and goes, okay, I know where I am now because there's a wall and so on. The magnetic sense, some animals use a magnetic sense for orientation. There's no evidence that rats and mice or humans do. So we think we're really visual. And even rats and mice who are not very visual for most things will actually mostly use vision to orient themselves. It's a really important sense for that because it's very precise. It's one of the most spatially precise senses. So that's very good.
when you were speaking about head direction and the importance to rats of sensing head direction, I found myself thinking of landing an aircraft in a crosswind. Because when you do that, you must go straight down the runway, but you mustn't be pointing straight down the runway. You've got to intentionally turn the aircraft to one side. So you're solving a really interesting problem in directional perception. And it occurred to me that's a process that must be true in a lot of these facilities. And I wonder if anybody's tried studying it without putting electrodes into pilots' heads.
I'd love to do that. Yeah, that's a really interesting question because something we haven't figured out yet. So I've been talking about head direction cells and they fire when the head is pointing in a particular direction and they're used for navigation. But you've made the point that you may be navigating, you may be moving in a direction that's not the direction that you're pointing in. And how does the system cope with that? And one study has tried to to separate out, are the head direction cells really interested in the direction of the head or are they actually interested in the direction of travel? And it's the direction of the head. So what's the bit of the brain that knows about the direction of travel? We don't know the answer to that yet. It's a really interesting question. So give us another 5 years. I've lost track of who's next. I'm ready for a change.
I've always been intrigued by animals, especially sea creatures, that can find their way back to, like for example, a sea turtle that can find its way back to a particular beach one night a year, but not just it's managing to navigate back to that, it's also got some kind of calendar in its head, or a penguin who goes off and feeds for 3 months and then comes back and shoots out the water at exactly the right spot, or whales that migrate for 2,000 miles to exactly. So there seems to be an enormous amount of other possibilities of how the brain is working, because those once you're able to see, they don't have the visual references at all. Has anyone explored how that's working?
Yeah, so there's two things. One is this type of really long range navigation that you're talking about. There's some, I think there's, I don't know if it's a consensus, but I think a lot of people think it's not quite the same. So some studies of long-range navigation in pigeons, for example, have found that they can do that fine without a hippocampus. But what they are disrupted in is when they get close to the loft, the home nest, then they get lost. So it looks like they're using something different to do the long range bit than they are to navigate around the familiar area with the landmarks. And so studies of some of these long-range migratory animals, that like different species use different things. Some of them use a magnetic sense. So there are, a lot of species do actually have this. Some of them use olfaction, as far as we can tell. Some things we don't know what they're using. It's just a mystery. There's a lot of very, very interesting work going on looking at these things. And yeah, the role of the hippocampus, we don't know. So there's some interesting work on semi-long range navigation that's being done by a group. So the scientist is called Naka Mulanovsky, and he studies bats. And he uses the same recording method to record place cells and grid cells and so on. But because they fly, he's able to track them over quite long distances. So he uses wireless recording and tracks. And he's found that you can get place cells that are like 1 kilometre long, like with the area where the cell will fire is like 1 kilometre long and so. So even the hippocampus over long distances for those animals is trying to make a very large scale map. But there's a lot we don't know. Like dolphins and whales, what do they do? We just don't know. Dying to know the answer to that. It's not that easy to find.
Has anyone looked at the brains of the whale or the turtle or? Yeah,
they have, and the brains. I mean, turtles are a bit different from us, because they diverged a long time ago in evolution. But whales and dolphins and things, they're mammals. They're not totally dissimilar to us in many ways. The brain architecture is quite similar. There are some differences, but they have hippocampus and all the rest of it. And so I'm sure they use them, the same things that we do. But But what they use them for in terms of their navigation, we still don't really know. But, yeah.
Is there any difference between male and female rats in there? But in particular, whether it's like they forage equally, therefore that sort of learning of position is far more appropriate, and the consequential groan from the audience that says that there's a much bigger overlap in human capability, but some seem to enjoy one versus the other.
Yeah. So it's been something that's been investigated quite intensively, and I think the answer's still not really clear. So the basic story is that if you look at males and females of rats and humans and some other species as well, that males are more likely to rely on a global sense of direction, and females are more likely to rely on landmarks. And so some of the evidence is quite interesting, but the sample sizes tend to be small and it's not very easy to replicate. And in humans, it's difficult to interpret the studies because male and female experiences are different. So, for example, males tend to drive more than females, so they're kind of trained in long range, kind of attending to global cues, if you like, because they're moving through. They're having to navigate at high speeds and so on. So it's not clear the extent to which these are innate. And so I've looked at sex differences in the virtual reality study, the map drawing that I've been doing. I've not seen any differences. But what I have seen, and this is quite well replicated, is differences between male and female mental rotation of small objects. So if you do the classic psychological mental rotation test, so you show somebody an object like a three-dimensional Tetris brick, and then you show them rotated versions or altered versions, and say which of the rotated versions and which are actually different. Males score slightly better than females, and it's a very reliable finding. I have no idea what to make of that. It's bizarre, but it's quite well replicated.
The corollary is, have you talked to the people who do map reading, mountain rescue stuff with regard to cues? The bits you were talking about.
Yeah, I don't know.
The same as for architects going, this will help you learn the stuff.
Yeah, but again, it's confounded by experience. So little boys tend to play with construction toys and all of that kind of stuff. So they do have different types of practice. So we don't know whether these are genetic differences or whether it's just kind of society. I mean, it's an argument that's not going to go away probably in my lifetime. But everyone is always interested.
Thank you.
Sorry. It's kind of following on, but it's more about neurotypes. Has anyone looked at the differences between autistic neurotype and because there's a high prevalence of people that are in spatial careers, things like sculptors or dancers or people that use their body a lot more. Is there a difference in the way they're creating the spatial map? The reason I ask is I'm very good in the real world, and I play a lot of computer games with someone who's really good in the real world, in the virtual world, but sucks in real world, and I'm the other way around. And so is there something about the body that is linking with it more so, just with different neurotypes?
Yeah, I don't know. I mean, there's a lot.
Sorry, I don't think most people would have heard.
Oh, okay. So is there a difference in neurotypes? People's, I don't know if I'm going to interpret this correctly. People's tendency to be able to manipulate things spatially in the real world versus the virtual world. These individual differences, and do those map onto neurotypes like autism? Is that fair summary? I don't know the answer to that. It's a really interesting question. I think the difference between the virtual world and the real world, that might come down to a difference in mental rotation, so imagination, and the ability of people to imaginatively rotate, which is something that I'm starting to get interested in. I think there probably is a literature on autism, but I'm not familiar.
And with your virtual thing, it was quite architectural, but it was making some of the same patterns that the architects that you're complaining about. So having things in there that people are grounded to might help.
Yeah, well, I was trying to remove those things.
Deliberately.
to see,
Right.
so that they couldn't use those, because I wanted to understand their ability to make maps just from tracking their movements around corners.
Right, so they're
Things like that.
physically moving or they joystick moving.
So just a joystick. That's using a joystick.
So I have actually always had quite a good sense of direction, you know. Certainly better than my wife, anyway. But I have a good sense of direction. And but occasionally I find myself getting lost. Like a couple of weeks ago, we were in Venice. Wall was there, and Venice is a maze, and I like to go wander around and get lost. Yeah. So when you get this feeling of getting lost, are there, now that we know how our brain is orienting itself, are there kind of strategies for orienting yourself again?
Yeah, I mean, the reason for getting lost, I think, is that there's not any really clear directional cues that are that are visible from multiple regions. So Venice is kind of parcelled up. You know, it's carved up by the streets and the canals, and so wherever you are, you can just see the local environment. But then you go around a corner, and especially because the canals curve and things like that. So it's very easy to lose your sense of global orientation, and then that makes it hard. And the buildings all look the same to me anyway. They all look lovely, but they all look the same. So I think it's confusing, but it's kind of a nice confusingness, I find. And then every now and then you get somewhere you recognise. You get to the square and you go, okay, now I know where I am. And it kind of resets your head direction system, can kind of knock on again. But I find my phone indispensable.
Thank you, that was a lovely talk. It was clearly very important what you're telling us. Have you been able to translate it to politicians and, you know, NHS Scotland, so that they actually start telling nursing homes where we have all these increasingly demented people that they do need to build in these directional cues, like you suggested. You know, one wall in every room is painted a certain colour. Have you been able to translate it, or are you just too busy doing the science?
No, there is a big enterprise looking at dementia and how this research and navigation can help people with dementia. And there are some architects who are particularly interested in designing for people with dementia. The problem is that they're losing the substrate for navigation. So if you can provide them with all this information, but their brains can't use it. And the other problem, I think, is that it's a progressive disease. So, you know, who are you designing for? Someone who's in the early stages or someone who's in the later stages? And I think the things that they're able to use change over the course of the disease. So I think I'm not sure that we are going to come up with a one-size-fits-all design strategy.
So we need it in our homes rather than in our nursing homes to stop us getting there.
Yeah, if we could stop people from getting dementia, that would be great. And I think you can. You know, I think it probably is possible to use non-spatial things. So I know that like care homes will often they'll paint the doors different colours, and you know, say you want the yellow door for the toilet or something like that. So we'll try and use something non-spatial. But these people are also then losing their memories, so they lose even the ability to. So I think really the philosophy for me that would make sense is to design for the carers to make life as easy as possible for them. And also, so one of the architects who's quite interested in dementia is this guy called Neil McLaughlin, and he designed this award-winning care home in Dublin that capitalises on the fact that people with dementia will often wander. And I don't know why they wander, but it's interesting that animals with damaged hippocampuses will wander as well, and so I suspect it might be because they don't recognise where they are, and so they're constantly moving. I'm going to keep going because I don't know where I am. And he designed this so that if they wandered, it would take them on a kind of interesting path, but it would take them back to the communal centre of the home. I just thought that was brilliant. So I think strategies like that probably will help.
Picking up on what you're saying about some of these neurons firing through a sense of awareness of the body moving. Ties up nicely with that perhaps the more you move, the more you're stimulating these cells, and that when you stop moving, you start to lose this mental mapping to a degree. Is that a possibility?
I think it is a possibility. And people are kind of looking into to that, the extent to which exercise, for example, seems to be really beneficial for the hippocampus. And for two reasons. One is that, like you say, moving around, moving through space is stimulating it. But also the physical act of exercise seems to stimulate this thing called neurogenesis, that's the birth of new neurons. The hippocampus is one of the only places in the brain that will make new neurons, and that can be stimulated. It's a little controversial, but I think it's becoming gradually clearer that can happen. So exercise is really good for brain health. You probably all know that. I know that, but I don't follow. I think you do too.
Sorry, couldn't see you there.
Hi, yeah, thank you for your talk. You seemed to suggest earlier on the talk about consciousness potentially being born out of matter. Maybe I picked that wrong, but and further on from that, it's like even at the point when you're a single cell, it could be argued that there's a degree of spatial awareness at single cell level. So therefore, how much of it is linked to it being merely a means of survival?
What do you mean by means of survival?
The spatial awareness, being able to kind of like the navigation, it's a survival mechanism.
Yeah, very, very much so. And I think that the evolution of the spatial awareness, and awareness is a bit of a fraught term, spatial processing, let's call it. I think that probably happened really early on, when in the very first animals started to be able to move around. They had some ability to detect things in their environment and move towards a way forward and learn that in the simplest way, and then build up structures that allowed that to become more and more sophisticated, and then that became the hippocampus. So the hippocampus seems to start a long, long time ago in evolution, and so we see a homologue of the hippocampus in birds and reptiles, and some people argue insects. I'm not sure I'd go that far, but there's a hippocampus-like thing in insects as well. So, yeah, very important for survival.
I'm just wondering, for humans, would you have different space cell marks depending on the speed you're following the route? And the reason I'm asking that is that I do a lot of cycling, and I'll learn a route so I know it really quite well, but when I'm in the car doing the same route maybe for the first or second time, it doesn't seem nearly as familiar to me. And there's the difference is the speed. And I just wondered if you create different space cell marks because of that.
Yeah, quite possibly. Yeah, quite possibly. So there's this phenomenon called remapping, which is if you change the environment, the place cells will make a new map. So if you change the colour, they'll make a new map. And one thing, so the experiment that I'm thinking of is from this bat lab, where they looked at bats flying or walking, and they have a different map for flying versus walking, or for using vision versus echolocation and so on. So it's same space, but different way of accessing it. So I think that's probably right, that you have a different map, and that's why it feels strange.
Intelligence, and because that seems to have, you've got a phenomenally complicated, you showed us your flowchart, bringing that all together. Are you using there, do you see research progressing much faster now and it's bringing these patterns together?
You mean because of AI? Will AI help?
Yeah.
So, I mean, AI certainly gives us interesting tools because, it's got this ability to find patterns in huge amounts of data. So we're definitely using AI to analyse the vast quantities of data. So, I talked about O'Keefe's experiments where he put an electrode into the brain. We now can put thousands of electrode recording sites in, and you've got so much data. And we use AI for that. In terms of AI becoming intelligent, like being able to model the brain with AIs, I think we're a long way from that yet, because we don't really know how the brain works yet. We're just at the very beginning stages. But it's certainly an exciting and very hot area of research.
I was just thinking, your research, do you see it sort of, there's so much you know and so much you've still got to find out whether that's going to increase or that rate of learning will become even more advanced than it is at the moment?
Yeah, I think so, so long as we keep getting funding for it, because it is expensive. And the brain scanners and recording systems, everything, it costs a lot of money and it takes a long time. And the complexity is such that we can do lots and lots of experiments, but our increment in real understanding is quite slow. It's really the most complex system in the universe in a way, and we don't have a good understanding of complexity yet. So, but I think it's going to be really interesting in the future.
And I was really interested to hear about your experience when you came to Edinburgh and you got lost, because I had the exact same experience when I went to Sydney. And I had a really good sense of direction, but when I found myself in Sydney, I was completely lost. And that's made me wonder about the magnetism effect, just simply being in different hemispheres.
I don't think it's magnetism, but one thing that people often say to me is when they go from the northern hemisphere to the southern hemisphere, they get confused because the sun goes the wrong way across the sky. And I think we don't really, here we don't really use the sun, right? Sun's just wrong. But if you come from a sunny place, you have this sort of instinctive understanding that if it's morning and you can see the sun, that's the east, and if it's the evening, that's the west, and you use that without realising it. And so when you're in the southern hemisphere and you're trying to do that, you're just getting it completely the wrong way around. So I've had a lot of people say that anecdotally. I'm not sure if there's strong evidence for it.
But did you, when you go back, do you retain your ability at mapping? Because I found I never regained the ability I had before I went south.
Interesting. Yeah, maybe you had just stopped using the sun. I don't know. Yeah. No, it can be just sort of disconcerting.
I don't know whether there's any proof of this, but in Dundee recently at the McManus Gallery, there was an exhibition on the impact of sound, sonic boom on whale speeching, and what you could do was put your hand on one of those things and feel their vibration. Now I don't know if that's but what it's making me think about is the impact of sound noise on our ability to orientate ourselves, because of, I'm at the stage now where people going to raves and discos and the noise is so loud. Do you know whether that could actually really impact on your sense of direction if you've got a heck of a lot of noise going on?
Yeah, I think quite possibly. We certainly can use noise, sound sources as one of the sort of sources of directional information. So if that's being masked, you've taken away something, but also just being exposed to sound is stressful, and stress has a short-term effect of impacting your attention to things. So if your brain is dealing with this really unpleasant stimulus, it's maybe not attending as much to kind of landmarks and so on. And then long-term exposure to stressful things actually raises levels of the hormone cortisol, which has damaging effects on the hippocampus. And so probably not in your example wouldn't affect, but stress, can be detrimental to the hippocampus.
Okay, maybe one more question. If anybody's got one. No? Oh,
I'd agree with the North Hemisphere, South Hemisphere. What he said, but we hear that Aboriginal children or Kalahari, that they are people basically who can find things, sources of water and so on, where the sort of non-indigenous people can't, and that I think research was stopped because of eugenics and so on. But it was an interesting thing. Is there any suggestion that there is actually any differences between races?
No, I don't think so in terms of the physiology, but there's a lot of cultural transmission of navigational knowledge, and it's really interesting to study indigenous peoples who are still using the old methods of navigation. So another example, there's the Australian Aborigines who go across the desert, but there's also the Polynesian navigators going across the ocean. And they have a kind of intergenerational transmission of knowledge. So the Aborigines have these things called song lines, which are kind of narratives. I'm not very familiar with it, but that they pass down from one generation to the next, that are pointing out the kind of salient places in the desert, that like kind of spiritual places or water places and so on and so on. And they pass these things down from generation to generation. So they have a kind of a route map, if you like, of where things are. And the Polynesian navigators will use things like their knowledge of the currents and the wind direction and things like, you know, the seabirds. If they could see seabirds, they know where they are in relation to the islands and all that sort of stuff. And that all gets passed down from generation to generation as well. So we humans, because we have language, we have this whole extra dimension that other animals don't have, and it's been really incredible for our development, because we can kind of reason. So we're not just using our instincts of knowing where things are, but we can reason logically as well, and then pass that on. So, you know, we're very different in that regard. And we can write things down.
I think we need to get Kate to the 9:30 train. So can I ask David Bowler to do a vote of thanks?
I think, like all of us this evening, I was very much looking forward to this talk, but for special reasons, because perhaps more than most people, I was brought up on maps. My dad was in the Royal Air Force during the war. His father was at sea. And my maternal grandfather had a remarkable talent for navigating the rural areas of Hampshire by means of a mental map of all the public houses between Easton and Southampton. There were experimental possibilities there that were never fully taken advantage of. But I think, and I also have, as a sort of legacy of my student days, a mental map of all the branches of a ladies' fashion outlet around the island of Montreal, from having worked with truck drivers delivering to all of them. So I think all of us in our different ways, and myself especially, would have been able to identify with what we were hearing this evening, and to, as it were, analyse all the mapping going on inside our own heads and how we do it. And this evening we've been given an insight into exactly what's going on. I'm sure we'll go on doing this, sometimes thinking about it, sometimes not. And when we do think about it, we'll think about it a lot more, thanks to what we've heard this evening, which I know we've all enjoyed tremendously. So thank you so much for coming, and thanks for being with us. Thank you.